You are most welcome to this talk. Now, today we are going to be looking at something that has generated considerable discussion in the medical literature, and I think it is important that we understand what the data actually shows and what it does not show. We are going to be examining the immunological mechanisms by which messenger ribonucleic acid vaccines interact with the human immune system, and specifically we are going to look at the published research regarding autoimmune phenomena that have been reported in the post-vaccination period. Now, before we go any further, I want to be absolutely clear about what this video is and what it is not. This is an educational discussion based on publicly available peer-reviewed research. I am not here to tell you what to do. I am not here to give you medical advice. What I am here to do is help you understand the data so that you can have informed conversations with your healthcare providers. The science on this topic is still evolving, and we need to approach it with appropriate intellectual humility. Right, so let me show you the background here. The human immune system is extraordinarily complex. It has to perform what is essentially an impossible task. It needs to recognize and attack foreign invaders, things like bacteria, viruses, parasites, while simultaneously leaving your own tissues completely alone. And the way it does this is through a process of immune tolerance. Your immune cells learn during their development to recognize self from non-self. They learn what belongs to your body and what does not. Now, autoimmunity occurs when this system breaks down, when the immune system mistakenly identifies your own tissues as foreign and attacks them. This is what happens in conditions like rheumatoid arthritis, where the immune system attacks the joints, or type 1 diabetes, where it attacks the insulin-producing cells of the pancreas, or multiple sclerosis, where it attacks the myelin sheath around nerves. These are serious conditions, and they affect millions of people worldwide. The question that researchers have been investigating is whether vaccination, and specifically messenger ribonucleic acid vaccination, might in some individuals trigger autoimmune responses. And this is not a new question. Vaccine-associated autoimmunity has been studied for decades across many different vaccine platforms. What we are looking at today is the specific data regarding the SARS coronavirus 2 vaccines. Let me walk you through the proposed mechanisms, and I want to emphasize that word proposed. These are hypotheses that researchers are investigating. They are not proven facts. The first mechanism that has been discussed in the literature is molecular mimicry. Now, molecular mimicry occurs when a foreign protein, in this case the spike protein produced by the vaccine, shares structural similarities with proteins in your own body. If this happens, antibodies or immune cells that are generated against the spike protein might also cross-react with your own tissues. This is not a new concept. Molecular mimicry has been implicated in various autoimmune conditions triggered by infections themselves. There have been studies, and I am going to show you some of the data here, that have identified sequence homologies between the SARS coronavirus 2 spike protein and various human proteins. A study published in Clinical Immunology in 2021 identified significant sequence similarities between the spike protein and human proteins involved in cardiac function, coagulation, and neural processes. Now, does this mean that the vaccine will cause autoimmunity? No, it does not. Sequence similarity does not automatically mean cross-reactivity. The immune system is far more sophisticated than that, but it does provide a theoretical basis for why some researchers are investigating this question. The second mechanism that has been discussed is the presence of autoantibodies in the post-vaccination period. Now, several studies have documented the appearance of various autoantibodies following messenger ribonucleic acid vaccination. A study from Germany published in 2022 looked at antiphospholipid antibodies, which are associated with a condition called antiphospholipid syndrome. They found transient elevations in these antibodies in a proportion of vaccinated individuals. However, and this is crucial, the presence of autoantibodies does not necessarily mean clinical disease. Many people have circulating autoantibodies without ever developing autoimmune conditions. Let me show you another study. Research published in the Journal of Autoimmunity examined sera from individuals who had received messenger ribonucleic acid vaccines and found the presence of anti-nuclear antibodies in some subjects. Anti-nuclear antibodies are associated with conditions like lupus. But again, the clinical significance of these findings is not yet clear. We need longer follow-up studies to understand whether these laboratory findings translate into actual disease. The third mechanism that has been proposed involves the lipid nanoparticle delivery system. The messenger ribonucleic acid in these vaccines is encapsulated in lipid nanoparticles to protect it and help it enter cells. Some researchers have suggested that these lipid nanoparticles might have adjuvant properties, meaning they might non-specifically activate the immune system in ways that could potentially trigger autoimmune responses in susceptible individuals. There is some experimental data supporting this hypothesis in animal models, but the relevance to human autoimmunity remains to be determined. Now, here is what is important, and this is really the key point of this video. We need to distinguish between theoretical mechanisms, laboratory findings, and clinical outcomes. Just because something is biologically plausible does not mean it occurs frequently or at rates that are clinically significant. Let me show you the pharmacovigilance data. The Vaccine Adverse Event Reporting System in the United States, the Yellow Card System in the United Kingdom, and Vigilance in the European Union have all received reports of autoimmune conditions following vaccination. This is publicly available data that anyone can access. Reports have included cases of immune thrombocytopenia, Guillain-Barré syndrome, myocarditis, and various other conditions. However, and I cannot emphasize this strongly enough, these are passive surveillance systems. They tell us that these events occurred after vaccination. They do not prove that vaccination caused these events. Correlation is not causation. This is fundamental epidemiology. People develop autoimmune conditions all the time. The background rate of these conditions in the population is not zero. So if you vaccinate millions of people, some of them will develop autoimmune conditions simply by coincidence, not because of the vaccine. What we need to look at is the comparative data. Are people who received the vaccine developing autoimmune conditions at higher rates than people who did not receive the vaccine? And this is where the research becomes more nuanced. Some studies have found signals. A large study from Israel published in 2022 using data from a major health maintenance organization found small but statistically significant increases in certain conditions in the vaccinated population compared to the unvaccinated population. These included herpes zoster reactivation and certain inflammatory conditions. The absolute risk increases were small, but they were there. Other studies have found different results. A study from the United Kingdom using primary care records found no significant increase in most autoimmune conditions following vaccination. A study from Denmark looking at inflammatory bowel disease found no association with vaccination. So what do we make of these conflicting findings? This is the challenge of real-world epidemiological research. Different populations, different methods, different definitions, different follow-up periods can all lead to different results. The honest answer is that we do not yet have complete clarity on these questions. Let me talk about myocarditis specifically because this has been one of the more clearly established associations. Myocarditis is inflammation of the heart muscle. Multiple studies from multiple countries have now confirmed an association between messenger ribonucleic acid vaccination and myocarditis, particularly in young males after the second dose. The mechanism appears to involve an inflammatory response, and some researchers have suggested autoimmune mechanisms may be involved. The rates are relatively low. We are talking about something in the range of 1 to 4 cases per 10,000 in the highest risk group, which is young males aged 12 to 17. Most cases have been mild and self-resolving, but this is an example where a safety signal was identified, investigated and confirmed through rigorous epidemiological methods. This is how pharmacovigilance is supposed to work. Now, I want to address something that I think is very important. There has been discussion about what has been called immune imprinting or original antigenic sin in relation to repeated vaccination. The concern here is that repeated exposure to the same antigen might train the immune system in ways that are not optimal and might potentially lead to immune dysregulation. There is some theoretical basis for this concern, and there is ongoing research examining immune responses in people who have received multiple doses of the vaccine. A study from Cleveland, published in 23, found that individuals who had received more doses of the vaccine had higher rates of coronavirus infection than those who had received fewer doses. Now, this study generated considerable controversy. Critics pointed out potential confounding factors. Perhaps people who received more doses were at higher baseline risk for other reasons. Perhaps there were differences in behaviour or exposure. The study authors acknowledged these limitations. The point here is not that the vaccine does not work or that it causes harm. The point is that these are complex questions that require careful scientific investigation. We should not dismiss concerns out of hand, but we should also not jump to conclusions without adequate evidence. Let me talk about what we know and what we do not know. This is really important. What we know with reasonable confidence is that messenger ribonucleic acid vaccines trigger immune responses. That is their purpose. That is how they work. We know that in some individuals these immune responses can be robust, even excessive. We know that myocarditis is a real, if uncommon, adverse event associated with these vaccines. We know that auto-antibodies can be detected in some vaccinated individuals. We know that there is biological plausibility for vaccine-associated autoimmunity through mechanisms like molecular mimicry. What we do not know is the precise incidence of vaccine-associated autoimmune conditions. The data are inconsistent across studies. We do not know which individuals are at highest risk. We do not have good biomarkers to predict who might develop autoimmune complications. We do not know the long-term implications of the immunological changes observed in some studies. We do not know whether the autoantibodies detected in some vaccinated individuals will lead to clinical disease over time. This uncertainty is uncomfortable, but it is honest. And I think honesty is more valuable than false certainty in either direction. Now, let me address the methodology issues because I think this is crucial for understanding the data. Many of the studies on vaccine safety are observational. They look at people who chose to get vaccinated and compare them to people who chose not to get vaccinated. But these groups are different in many ways. People who get vaccinated tend to be different from people who do not in terms of age, health status, healthcare seeking behaviour, and many other factors. These confounding variables make it very difficult to establish causation. The gold standard for establishing causation is the randomized controlled trial. And the original trials of the messenger ribonucleic acid vaccines did show that they were safe and effective. But these trials had limitations. They were not powered to detect rare adverse events. The follow-up periods were relatively short, and in many cases the control groups were subsequently vaccinated, making long-term comparisons impossible. This does not mean the vaccines were not properly tested. It means that rare adverse events, particularly those that might take months or years to manifest, are very difficult to detect in pre-authorization trials. This is why post-marketing surveillance is so important. And this is why we are having this discussion now, because the post-marketing data is accumulating and needs to be honestly evaluated. I want to share some perspective here. The human immune system is remarkably resilient. Most people who receive vaccines, including messenger ribonucleic acid vaccines, do not develop autoimmune conditions. Even among those who develop transient autoantibodies or inflammatory responses, most do not go on to develop clinical disease. The immune system has multiple regulatory mechanisms to prevent autoimmunity, and in most people these mechanisms work effectively. At the same time, we know that some individuals have genetic predispositions to autoimmunity. We know that environmental triggers, including infections, can precipitate autoimmune conditions in susceptible individuals. It is biologically plausible that in some genetically predisposed individuals, vaccination might serve as a trigger. This does not mean vaccination is unsafe for the general population. It means that individual risk assessment is important. This brings me to the question of what you can do with this information. And again, I am not going to tell you what to do. That is not my role. What I would suggest is that if you have a personal or family history of autoimmune conditions, this is information that you should discuss with your healthcare provider when making decisions about vaccination. If you develop new symptoms after vaccination, these should be reported and investigated. If you have concerns, ask questions, you have the right to informed consent, and informed consent requires information. The regulatory agencies continue to monitor the safety data. The World Health Organization, the European Medicines Agency, the Food and Drug Administration, the Medicines and Healthcare Products Regulatory Agency in the United Kingdom, all of these bodies have ongoing pharmacovigilance programs. When signals are detected, they are investigated. When risks are confirmed, guidance is updated. This system is not perfect, but it is the best system we have. Let me conclude by saying this: science is a process, not a destination. Our understanding of the immunological effects of messenger ribonucleic acid vaccines will continue to evolve as more data accumulates and more research is conducted. What was considered definitive knowledge 2 years ago may be revised as new evidence emerges. This is not a weakness of science, this is how science works. We should be suspicious of anyone who claims absolute certainty on these questions in either direction. The questions about vaccine-associated autoimmunity are legitimate scientific questions that deserve rigorous investigation. Dismissing these concerns as conspiracy theories is unhelpful. Exaggerating these concerns into claims that vaccines are universally dangerous is equally unhelpful. The truth, as is so often the case, lies somewhere in between. There are signals in the data that warrant attention. The magnitude and clinical significance of these signals remain to be fully determined. I encourage you to stay informed, read the primary literature if you can, look at the actual data, not just the headlines, be skeptical of claims that seem too dramatic in either direction, and most importantly, have these conversations with qualified healthcare professionals who can help you interpret this information in the context of your individual health situation. This video is for educational and informational purposes only. It is not medical advice. If you have symptoms or concerns, please consult a qualified healthcare professional. We use publicly available data and reputable sources, and interpretations may evolve as new evidence emerges. Now, before I finish, let me address one more aspect that I think is often overlooked in these discussions. We need to consider the immunological consequences of natural infection as well. SARS coronavirus 2 infection itself has been associated with autoimmune phenomena. Long coronavirus, which affects a significant proportion of infected individuals, appears to involve immune dysregulation in at least some cases. Studies have documented the presence of autoantibodies in patients recovering from coronavirus infection at rates that in some studies exceed those seen following vaccination. This context matters when we are evaluating the risks of vaccination. We should not be comparing vaccination to some theoretical state of perfect health with zero immune stimulation. We should be comparing it to the realistic alternative, which for most people is eventual exposure to the virus. And there is evidence, though not conclusive evidence, that the autoimmune risks from natural infection may be higher than the risks from vaccination in most individuals. A study from the United Kingdom published in Nature Medicine looked at a range of autoimmune conditions and compared their incidence following vaccination versus following natural infection. For most conditions examined, the risk was higher following infection than following vaccination. This does not mean vaccination carries zero risk. It means that risk assessment requires comparison to realistic alternatives. I also want to mention something about the immune system that I think is often misunderstood. The immune system is not a binary switch that is either working correctly or attacking your body. It is a complex regulatory network with multiple levels of control. Even when autoantibodies are present, even when there is some degree of self-reactivity, the regulatory mechanisms usually prevent overt autoimmune disease. The presence of autoantibodies in a laboratory test is not the same as having an autoimmune disease. This distinction is important. Furthermore, let me say something about individual variation. We are not all the same immunologically. There are genetic polymorphisms that affect immune function. There are differences in the composition of our microbiome that influence immune development. There are environmental factors, previous infections, nutritional status, stress levels. All of these things affect how our immune systems respond to any challenge, including vaccination. This individual variation means that population-level data, while important, does not tell us everything we need to know. Two people can receive the same vaccine and have very different immune responses. This is why personalized medicine is so important. This is why the conversation with your healthcare provider matters. They know your medical history. They know your risk factors. They can help you interpret general data in the context of your specific situation. Let me also mention the question of dose and timing. There has been discussion about whether the initial dosing regimen, which was established based on the pre-authorization trials, is optimal for all individuals. Some researchers have suggested that longer intervals between doses or adjusting the dose based on individual factors might reduce the risk of adverse events while maintaining efficacy. This is an area of ongoing research. The initial recommendations were based on the best available evidence at the time. As we learn more, recommendations may be refined. I think it is worth reflecting on how far we have come in our understanding of these vaccines. When they were first authorized, we knew they induced antibody responses and reduced symptomatic infection in the short term. We now know much more about the durability of immune responses, about the effectiveness against different variants, about the rare adverse events, about the immunological mechanisms involved. This accumulation of knowledge is valuable. It allows for more informed decision making. The challenges we face in understanding vaccine immunology are not unique to these particular vaccines. They are challenges inherent to studying complex biological systems in diverse human populations. Perfect information is not achievable. What we can achieve is progressive refinement of our understanding through careful research and honest evaluation of the data. I would encourage anyone watching this video to approach these questions with an open but critical mind. Be willing to consider evidence that challenges your prior beliefs, whatever those beliefs might be. Be skeptical of simple narratives that paint complex issues in black and white terms. The immune system does not deal in black and white. Biology is shades of grey, probabilities, and individual variation. The questions about autoimmunity and vaccination are important questions. They deserve serious scientific attention. They are receiving that attention from researchers around the world. As that research continues, our understanding will deepen. We should welcome this process, even when the answers are not as clear-cut as we might like them to be. Thank you for watching. Stay well, stay curious, and I will see you in the next video. I want to tell you about something that happened last week, something that nobody in Brussels wants to talk about.